Good you?
I'm good, thank you.
Okay, so I came by, I had some questions about last week's lab that we were doing about SDS Page and turbo transfer.
Sure.
I was a little bit shaky on what exactly does SDS stand for?
SDS, it's a sodium dodecyl sulfate. It's a compound that we use in the lab, and it will apply a negative charge to all the proteins that are present in your mixture, in your solution. And by that consequence, with the heating, if you remember we heat in the water bath, the heating will denature the protein, making it a primary structure, a linear chain. And instead, the SDS page will apply a negative charge. So the only variable that will differentiate your solutions, the protein in your solution, will be just by size, the molecular size of your protein.
Okay, so you said that it applies a negative charge to the protein, but what does that have to do with electrophoresis? Because like we know that it goes from negative to positive, but why is the SDS so important for this procedure?
So you'll be using a specific gel. The page is the polyacrylamide gel electrophoresis, and in that gel you'll have some gaps through which the protein goes through, and it will, as you said, it goes from negative to positive. And since all the proteins are negative, it will just stop by size. The only variable that will differentiate it is the size of each protein, and smaller will go through further the gel, while larger will stop before. If you want, look at this model that I have. So the model, it's used to determine SDS page. It shows SDS page, and it's used to determine which fraction contains the purified protein and understand the progress of purification from one fraction to the other. As you can see, imagine that the wood part, it's a chamber, while the toothpicks represent the gel containing the space for the protein to go through. On the side here, you have your control with known proteins that will stop through the gel at different molecular weights that you know. Instead here, you have the progression of purification from each fraction. This represented the solute fraction, this represents the wash fraction, this is dilute, and this is the purified protein. As you can see, the less purified contain more proteins with different molecular weight that stop via the gel. Instead, going through, we get more purified protein until we end up with our only recombinant protein, DHFR, that we can assume it's that one because the molecular weight stopped exactly where we know from our control.
All right, yeah, so that makes sense. And I remember mentioning the Langley buffer and its role of denaturing the protein further and also allowing for imaging when it's introduced to the molecular imager and when exposed to UV light.
Yes, yeah, exactly right. The Langley will help providing that linear chain of the protein, and also it will provide that blue color to visualize the bands in the gel, also with the molecular imager and the UV light after when the gel electrophoresis is done.
Could you show me some of the materials and things that we need in order to
Sure.
do this? Okay.
Let's go. Choose. Okay, so yes, this is the gel electrophoresis machine, and here it's where you're gonna be placing your gel, and it's gonna be standing here in a vertical position. The gel will contain little wells in which you will load your solution, your fraction containing the specific solution with the Langley dye. And once it's loaded, you'll go for the next one. You will add a specific buffer that will create with ions that will help to conduct the gel from negative to positive. And you will plug it. You will plug the machine that with a specific voltage will run for a specific amount of minutes from negative to positive, as you said, and will differentiate the solution by size.
It's kind of hard to load the samples into the gel, right? Because the wells are so small.
Yeah, that's the hardest part, and where students make usually most mistakes. That's why you will be using specific white tips that are really small that will go in the well, and then you have to have a steady hand. Usually we'll get in this position to hold it real steady, and you will slowly load the solution into the gel, until you see it, and thanks to the Lamley dye.
Mm-hmm. So then I remember from class, then the lid just goes right on top after everything's loaded. You plug the cathode and the anode in, and then run the gel. So
Yes,
that
exactly.
that makes sense.
Positive, positive, negative, negative. You run at specific voltage, and the gel will move towards the positive charge.
And then after it's done, how do we? So we crack this open and get the gel, and then we bring it where?
So yeah, you will crack the gel out, and then you will stain the gel with Comasi dye that will allow to visualize the gel with the molecular imager, and then we'll place the gel stain in the imager, and we'll visualize it with the specific program.
Okay. Do you want to go see the UV imager?
Sure.
Okay.
So this is the molecular imager. Here you will place your gel with a little bit of water, and once placed, you will close it, and the machine will read it, and you will be able to visualize on your computer on a specific program. You'll be able to visualize your gel for interpretation.
Okay, so again here, so this would be one of the results that we had from class, and you can kind of see in that first one where all the red is, that would be our molecular weight, the thing that we're measuring against. And then as you move on, you have the uninduced, induced, the soluble, the wash, elute, and then finally at the end we have our purified protein, correct?
Yes, absolutely correct. Imagine this, refer again to the model with the toothpick, and here you have your control marker with all the molecular weight of the known proteins, and then this, we're going through the progress of purification until here you get in the last one, a single band, if you can see it here, where this is the position of your DHFR, your protein that you try to purify, and this, we can assume that it was performed correctly, as the positioning comparing to the marker is around 52 kilodalton. That is the known molecular weight of our protein. So we can assume that this is a correct procedure, but we will have to verify later with the Western blotting.
So for Western blotting, I know that we have to perform the turbo transfer in order to transfer this protein of interest into a membrane, correct?
Yes, exactly.
And so when we do that, we're going to make a sandwich basically of the iron reservoirs, the cathode, the anode, the membrane, even the gel. Do you have a good picture or something?
Yeah, look, check out this picture that will explain really well. Over here, you can see it. Basically, the goal is that the turbo transfer will allow you to transfer the gel to a membrane, as the membrane is more stable for Western blotting later. And here you will be performing a sandwich. As you said, you can see the top cassette, the negative, with the iron reservoir stacked. And this will allow you, through an electrical force, to move down the gel, the proteins in the gel, to the membrane from a negative to positive again, as at the bottom of the sandwich you have the positive cassette and the positive iron reservoir stacked.
And so the proteins are still going to move from negative to positive, similar to how they moved in gel electrophoresis, because they're still coated in that SDS?
Exactly, yes.
Okay.
The SDS is still present and the negative charge is still present.
Okay, interesting. Do you have some of that material, the turbo transfer, so we can go see it?
Yes, I can show you.
Okay.
So this is the turbo transfer machine. Here, this over here is the negative cassette, where you'll be placing it on top of it with inside the gel first, the iron reservoir first, gel, membrane, second iron reservoir, and then at the bottom it's the positive cassette. You will close it, lock it, and then you will insert it in the machine, and then set up everything, all the function, and this will perform the transfer already by itself due to the electrical force to the membrane.
With some of the membranes, though, I remember we had to use the buffer, and we had to wash some of the membranes with the buffer. And could you explain?
Yeah,
that?
I mean, again, as in the gel electrophoresis, you have to add the buffer inside of it, and not the membrane, but the iron reservoir has to be washed in the buffer to allow this transfer due to the electrical force from the negative to the positive.
And then you just put it right here in the top slot here, run the program, and then after that it would be in the membrane?
After that, it will be in the membrane, and it will be good to go for Western blotting.
Okay, awesome.
So there's no real interpretation of result, as you have just to transfer from the gel to the membrane, and then Western blotting will confirm if it went through well or not.
Got it. All right. Well, thank you very much for your
Yep.
time. I appreciate it.
No problem.